So, what I'm going to have you guys start out with is just for each of these different tests, I've listed what they are. The first one is a lactose phenol red, second is methyl red, third is oxidase, fourth is indole. Front and back of these sheets. What I want you to do on your own with a partner, you choose what's best for you. Spend time looking at what type of metabolism is this actually testing for, so not what microorganisms is it differentiating, but specifically what type of metabolism. And then what are the results of interpretation? So for example, methyl red, we have two results up there. One obviously is negative, one is positive. You identify which one is which, and then what does that actually mean? Okay? I'm going to give you about 10 to 15 minutes to work on this. I don't want to rush through it. I want to make sure you have time to think about it before we report out on it. So that's all I've got to say right now. Any questions? All right, go ahead.
I got it turned down. Thanks.
In this case, the colors are a little bit different than what we see. That just probably means their water is a little bit more acidic than what we used to start out with, but not a big concern because they've got an uninoculated control, and then they have three others: E. coli, Shigella sonii, and Pseudomonas aeruginosa. What can we say about Pseudomonas? Negative. What carbon source are we testing for fermentation of here? Yeah, and that's one thing I wanted to make sure. This isn't saying that glucose is fermented. It's whatever carbon source is listed here. So the only sugar or carbohydrate within these tubes is lactose. So an organism could be positive for glucose fermentation, but negative for lactose fermentation. You may find actually tomorrow that that's true for your own unknown. Okay. That microbes have ability to ferment those different ones. Regardless of which carbon source it is, the results and the interpretation of these tests are very similar. You're focused on color change to indicate a potential acid being produced, and what else are we looking for? Gas production. So we know that Pseudomonas, no color change, it's unable. There's no acid, it's unable then to ferment lactose. What about the other two? Are they capable of fermenting? Yes. Which one is most definitely a heterofermenter? E. coli. What's the distinguishing feature there, Ashley? Yeah, so we said heterofermenters will produce more than a single product from their fermentation. And because not only we detect acid, but we also detect a gas here, that indicates two potential products. Therefore, E. coli we would consider to be a heterofermenter. We can't say with certainty, though, that Shigella is a heterofermenter of lactose because it may be that the acids it's producing are different types of acids. It could be producing lactic acid and acetic acid, or lactic acid and succinic acid at the exact same time. We just don't know unless we were to test further. Okay. Questions on this particular test? So the big things that I'm expecting you to know for these, and this is objective 4.5. It's being able to analyze and interpret these tests. So analyzing meaning, is there a color change? Is it positive or negative? And then the second part being, what does that actually mean? Ferments lactose in this case. Oftentimes in terms of workflow, methyl red comes after we work with phenol red. Why is that? What does methyl red actually test for? Or what is it determining? You're mumbling, but I'm kind of deaf today, sorry. Okay, types of acids, right? And what's the tipping point for methyl red? When does it exhibit its color change? 4.5, 5, somewhere in that range. Let me show you a visual that might be helpful here. So here is a graph. I pulled this out of a lab book, not ours, but another one. So in this particular graph here, what we have is pH value on the y-axis, hours of incubation on the x. In this case, the methyl red, the media for MRVP is a glucose-containing broth with some phosphate in it to buffer the solution. And these are showing, in both cases, we've got a fermentation occurring, but MR positive species in this case would be red. They're saying below 5. Above 5 would be ones that are MR negative, and most likely they are VP positive. And note in both of these cases, though, the pH is still below what? It's still below 7. And so what that would indicate is that an organism is capable of being phenol red positive. It's positive for fermentation, but yet negative for the methyl red. The methyl red is really testing for, of all the acids that are produced, it allows the stable acids are going to have a pH that's below 5. And these are going to be ones like lactic acid and acetic acid. The unstable ones, organisms will break down to form butanediol and acetylene. I'm not worried about you knowing those products. What I'm worried about you guys knowing is, do the acids that get produced by the organisms, are they stable or unstable and get broken down further? Okay, that's all I'm concerned with. Which of these is a positive result here? We've kind of covered this, but I want to make sure we hit it. Yeah, the red is a positive result here. This can sometimes be confusing because a red for phenol red test is indicative of a negative result. So it's just important to keep those two straight.
So would that mean that it's a stable acid?
Yes, yes. So the red would indicate that it's a stable acid that's being produced. Questions on this one? Okay. Let's go to our last bit. Oxidase and indole. What does oxidase test for? Derek. Okay, so whether the organism has cytochrome C oxidase, where do we find that? In the plasma membrane, part of what metabolic pathway? Electron transport chain. So that narrows it down. That tells us if an organism has this, it's capable of performing respiration. But what type of respiration? Aerobic. We associate this with aerobic respiration. This one is a really goofy test because it's a really nice indicator test. It's quick. It's easy to do. Usually I tell people that if you see cytochrome C oxidase, what this means is that the organism is most likely only going to perform aerobic respiration, that it's incapable of performing anaerobic respiration or fermentation. There are exceptions to this. There are always exceptions in microbiology and biology in general, but that's kind of a general theme that I tell folks. Which one of those is positive? Yeah, the dark purple one. The one on the right, people might say like, it looks positive. My guess is this test was probably exposed for too long. Not that I'm expecting you to memorize this for the exam, but the oxidase one was the one where I said, like, if you let it go for more than a minute or so, you will get false positives. So you have to be kind of careful in when you interpret the results for this test. All right, so in terms of metabolism, aerobic respiration, whether cytochrome C is present, which is part of that aerobic respiratory electron transport chain. Last one, indole. What is the indole test? Metabolism of tryptophan, ability to break it down. And what gets produced, Dana? Indole and anything else? We didn't really talk about what else gets produced. Not necessarily. The SIM test, where I think you're probably getting that from, the SIM test would test for sulfur or hydrogen sulfide production as well, but those two tests are actually independently of one another. They're occurring in the same tube, but you can have organisms that are H2S positive but indole negative, or vice versa as well. So it doesn't have to go hand in hand. When tryptophan gets broken down, we'll spend a little time with this one. But when tryptophan, which is an amino acid, gets broken down by the enzyme tryptophanase, a couple of products get produced. One is the indole. That's what we actually detect from this process. Another is ammonia. And the third product is one we know, pyruvate. So in this process, the breakdown of tryptophan, we get three products. What happens to that pyruvate? Or what could potentially happen to that pyruvate? What's that? Krebs cycle, exactly. So what we find is that in this case right here, in order for that tryptophan then to lead to some sort of energy production to provide ATP, that pyruvate has to go through respiration. Basically, by using tryptophan as a carbon source here, the cell has kind of bypassed using glycolysis altogether. Therefore, the reduction of pyruvate to lactic acid would yield no energy whatsoever. In order to get some energy out of it, pyruvate's got to go through Krebs cycle, give 2 ATP, and then go through electron transport chain. So what this says is that there are certain carbon sources, amino acids being one class of them, that can't be fermented. They can only be metabolized by some form of respiration, aerobic or anaerobic. Other examples of this are citrate. Citrate's one of the first products that gets produced within the Krebs cycle. When it gets brought into the cell, it actually gets converted, if I'm not mistaken, to pyruvate or acetyl-CoA, I can't remember which one, and then we'll go through the Krebs cycle. So there are a number of carbon sources that meet this demand, where if they are provided to a cell is capable of metabolizing them, however, only through aerobic respiration, or anaerobic respiration, sorry. And we've seen a couple of examples of that in lab. Trying to think if there's anything else I wanted to cover with that. No, I think that's about it. Questions on these 4 types of metabolic tests, Ashley?
So for indole, is it anaerobic or aerobic?
I think the best thing to say would be it's testing for Actually, I'm going to take that back. It's testing for whether the organism can break down the tryptophan. Yeah. That's really what it's testing for. But in terms of what happens with that tryptophan after it's broken down, in order for a cell to be able to metabolize it, it has to be respiration. And this really doesn't differentiate between whether it's anaerobic or aerobic respiration. It just tells us respiration, period. Yeah.
Is the right 2 on methyl red, would that be negative and on?
Yeah, The right tube, yes. So, sorry, I'm looking at the indole test. Yes, so the right tube on the methyl red, since it has that yellow color, pH is above 5. What that would indicate to us is that those acids, the acids produced during fermentation were unstable and have been broken down. Exactly. Meg? Which test was positive for indole? Yeah, it's the red one is positive. And this one in the yellow then was non-reactive. Thanks for asking that one, Mike. Any other questions? So, where do these show up on the exam? Well, there's a dichotomous key. I made it yesterday. It was so much fun. Actually, it was kind of infuriating. But there's a dichotomous key. You will not have to draw one, but you'll have to interpret one. And there are some test results on there where you will have to be able to determine, is this a positive or negative? What does it mean? Okay? So kind of keep those metabolic pathways in mind as you're thinking about this. Make sure you understand the interpretation of, or the results of these ones and whether it means it's positive or negative. That's the big thing. And this kind of takes care of that objective 4.5 again, which is analyze and interpret metabolic tests. Kaylin.
Are these the 4 tests that we need to know for the exam, or do we need to know all of them?
No, these 4, and there's one not on here, but we've talked about it so many times now, gram staining. You need to know gram staining. What's a positive, what's a negative, what does that mean in terms of the envelope structure, okay? But that's it then. Other questions? Okay, let me see what else I had planned, because I actually don't remember anymore. Okay, I'm going to make a couple of quick announcements, and then what we'll do is give you one review problem. So the first one is, don't forget, review session tonight, this evening, late afternoon, in here, 4:30 to 7-ish. I'll hang around till no later than 7. Come as you want, leave as you want, sleep in the back, whatever you prefer, eat as you want, etc. Second thing is your dichotomous keys are due tomorrow, so don't forget to either bring a hard copy or hand those in on Blackboard. 1 per group, so only one of you needs to submit it. I'll make sure that it doesn't count the other partner as late if they haven't submitted something. Third thing, start cultures today. Don't forget that what I need you guys to do for lab tomorrow is I'm going to make sure that there's nutrient auger plates and nutrient broth up in the lab. Your plates are already lying out though. They're just, you and your partners are stacked on top of each other and they're laid out by lab section. So you or your partner grab one of the plates. I need you to just do a T-streak for a new auger plate so that we've got fresh culture to work with on the plate, and then a fresh broth culture as well. And I will just put a test tube rack for each lab section in there so that you can just put them right into the put them right into that test tube rack, and then I'll put them where they need to go in order to grow. The plates you can just stick right into the incubator. And the lab's open all day today. There's nobody in there. Even if I'm not around, feel free to hop in and do what you need to do. And use any lab space because I'm not sure when people are going to be coming in. So, any questions about that? So just one of each. Make sure you label with your initials. And probably lab section would be helpful for me too when I get them, when I'm taking them out tomorrow. And then last thing is, I know about half people have done this already. If you want to fill out that survey for me to figure out who to place you with for labs, or not for labs, for lectures, let me know. I've started kind of grouping together, and just based on what I've seen so far, that's all. Okay. Any questions? Any questions about Wednesday? I want to make sure that I address any questions about the exam on Wednesday real quick too. Again, I'll be here at 7:45, so you can show up anytime after that to begin if you'd like. And maybe what we'll do is we'll do first row, third row, fifth row, seventh row, and then the same thing on the sides too. Okay, so the question again, 2 cultures of facultative anaerobe, same organism, growing in the same type of medium. One's exposed to air, the other's maintained under anaerobic conditions. Which of the 2 will contain more cells at the end of the week? Any guesses? What was that? The aerobic one? Anybody agree with that? Okay, Austin, why?
Because it wasn't capable of performing anaerobic. Aerobic still generates greater yield. So
Greater yield of what? Perfect. And so we said, and why is that? Anybody remember why anaerobic doesn't give as much, Ashley?
Because oxygen is more electronegative, so it allows for like a stronger pull in the electron transport chain.
And what does that allow for? Is those electrons pass through more of the more
Hydrogen
complexes?
goes out, and then
Good.
go through.
Good. So as more electron in aerobic respiration, as Ashley pointed out, oxygen's the most electronegative of all those electron acceptors we've talked about. So it allows for the most amount of energy to be released from those electrons as they pass from one complex to the next. That allows for the most hydrogen ions to pass across the membrane and allows ultimately for the most passive diffusion of those hydrogen ions back into the cell. Great. Could this organism perform fermentation? Possibly. It doesn't really say in there, right? I'm not even defining for you necessarily what's in the media. But fermentation only gives us how many ATP per glucose? Only 2. So we might find that like within 12 hours, the one growing anaerobically might have more as it's fermenting and growing and using up that glucose really quick. But at some point, if it were a fermenter, it might end up exhausting all of that carbon source and now having to switch its form of metabolism to anaerobic respiration, for example. That's kind of much more complex. I wouldn't expect that in the answer, but between Kaylin, Austin, and Ashley, that's a good answer. Okay, that would get you the points. Real quick recap on what we talked about from Friday. So, what are some differences between anaerobic and aerobic respiration? Okay, so final electron acceptor differs, right? O2 for aerobic, and then we've talked ad nauseum. Sulfate, nitrate, carbonate, 3 examples of final electron acceptors in anaerobic respiration. What else is different? ATP yield is different, so again, I'm just going to put this number not for you to memorize, but more for my purposes so that we can make that point. 38 ATP here, somewhere between 4 to 38 ATP. Yes, it's a broad range. What else? Those are kind of the major ones, right? And again, this difference here in the ATP yield is caused by these different electron acceptors and how much energy gets released from those electrons. Similar in what aspects? Good. So both if we're talking about breakdown of glucose, glycolysis, Krebs, ETC. ETC features what type of phosphorylation? Very good. And glycolysis and Krebs? Substrate level. Sublevel. Oxidation of the carbons from glucose? Complete or incomplete? Complete, and completed when? Krebs, in what form? What does carbon look like? CO2. Beautiful. Beautiful. Final electron acceptor for fermentation? Pyruvate. Gosh. All right, good. If you feel comfortable with those, I think you're in pretty good shape for those types of questions on the exam right now, okay? So again, that exam is covering everything from macromolecules all the way through what I'll call it stops right now, 8:46 on Monday. Take a couple of deep breaths. Take those practice exams. Keep working on some problems. Yes, I know you have anatomy and physiology too, but this is way more important than anatomy and physiology, and it always will be, okay? You can take that one to the bank. All right. Have a great rest of the day. I'll see you guys around today, tomorrow.